The following is about the Meselson and Stahl DNA replication experiment. Shortly after the Watson and Crick model of DNA was figured out and people understood DNA better, it did not take very much time before people wanted to understand how DNA was replicated. There were three main hypotheses for how this was done. The first hypothesis was that DNA replication was conservative, where the original strand is maintained and an exact replica is made. The second one is dispersive, which is where you have part of the original and part of a new strand mixed together. And there was a semi-conservative model which had part of the original strand and part of a new strand mixed together. Meselson and Stahl created an experiment to figure out how DNA replication was carried out. They grew E. coli on heavy nitrogen-15 and then they centrifuged it. After this, they saw that all of the bacteria went into the bottom of the flask. They then transferred it onto lighter nitrogen-14 and let it replicate and centrifuged it. And what they saw was that all of the bacteria DNA was in the middle. This disproved the conservative model because it would be expected that you would have one strand of heavy DNA and one of the lighter DNA at the top instead of one in the middle. But they let it replicate again on the nitrogen-14 and centrifuged it and saw that they had one band in the middle and one band of the lighter DNA at the top. This showed that it was semi-conservative because if it had been dispersive, you would have still seen only one band in the middle. So, the conclusion of the experiment was that DNA replication is semi-conservative, where the original strand is maintained and uses a template to create another complementary strand. Half of the newly created DNA strand is new, while the second half is old. So, with a simple experiment using just a few replications, Meselson and Stahl were able to show that DNA replication was not conservative, that it was not dispersive, and that in fact DNA replication is semi-conservative.